Hello, my name is Mohsin Beg, and I did my research project on exploring high CBD Rick Simpson oil as a viable treatment for multiple sclerosis and its consequential symptoms in adults aged 18 to 65. The cannabis plant has been documented to have strong medical properties for thousands of years. Modern science has allowed us to gain a better understanding of certain chemical processes induced by the use of cannabis and its related products. Cannabidiol, or CBD, a major component of cannabis, has been found to induce a variety of positive therapeutic effects upon the body. For this research study, the question is, can high CBD Rick Simpson oil, a concentrated oil extracted from medical marijuana, help treat clinical symptoms of multiple sclerosis seen in patients who are aged 18 to 65, in comparison to using conventional allopathic treatment methods to alleviate symptoms of MS? Can CBD RSO provide a greater degree of therapeutic relief for the patients suffering from MS and even possibly prohibit the further neural degeneration typically seen in the neurological disease? In order to understand the rationale behind the purpose of the study, one must have an understanding of the various mechanisms of multiple sclerosis as well as the mechanisms behind the consumption of cannabis products. If this study produces successful results, then modern allopathic medicine can manufacture drugs containing certain forms of CBD molecules to help alleviate symptoms of MS and various other diseases and illnesses. Multiple sclerosis, or MS, is an autoimmune disorder in which a patient's immune system starts to degenerate the myelin sheath, which encases various nerve fibers across the body. Deterioration of the myelin sheath causes damage to the given nerve fiber, leading to a large variety of different side effects or complications for the patient given the location of the nerve fibers affected. Areas where autoimmune damage has occurred to the myelin sheath can develop very different forms of lesions, abscesses, as well as localized inflammation in the central nervous system, which can be easily detected by an MRI exam. As a result, facilitation of nervous transmissions can hinder signals from the central nervous system to the peripheral nervous system, leading to a variety of complications for the patient depending on location of the affected myelin. Some of the difficulties MS patients may face may range from harmless tingling sensations throughout the body, all the way to paralysis and inability for normal bodily functions to occur. In some cases of MS, the severe cognitive effects may cause economic, psychosocial, and physiological implications affecting the overall lifestyle of the affected patient. Cannabis has two main compounds which are thought to have medicinal value. They are tetrahydrocannabinol, which corresponds to the activation of CB1 receptors in the body, and cannabidiol, which activates CB2 receptors present in the body. Medical marijuana has been noted to have many positive effects on the health of individuals suffering from various forms of ailments. The human body contains special cannabinoid receptors within the nervous system which accept the molecules from cannabis as if it were a puzzle piece fitting into another corresponding piece, resulting in the activation of the body's cannabinoid receptors. Many research studies suggest that activation of these cannabinoid receptors within the body causes a direct effect on the human body's immune system, with the tendency of having high levels of therapeutic potential for individuals suffering from diseases involving the body's immune system, including MS. A study published in the Scientific Reports analyzed the potential of immunomodulation from the oral administration of cannabis in conjunction with lipids to rats within a lab environment. The study justifies the test on a small animal such as a rat, since levels of cannabinoids will be much higher ratio in the body of a small mammal, therefore giving much more prominent results which can then be researched and easily analyzed through the results that they obtained. The methodology implied within the research study included running two sets of controlled trials in which one group was exposed to cannabinoids while the other was the control group. Researchers found that cannabinoids had biodiffused into the lymphatic system of the rats, especially the cannabinoid CBD, which was found to have high levels of proliferation within the lymphatic system in comparison to THC. Results of the study suggest that cannabinoids within the lymph system suppress various cell signaling proteins which are responsible for inflammation and pathogenic marker cells of autoimmune diseases, causing overall positive immunomodulative effects. Another study analyzed the potential of the specific cannabinoid CBD for its possible therapeutic effects on the autoimmune mechanisms within the body. Similar to the study previously discussed, this study also used animals, mice in particular, to conduct their research on the effects of cannabinoids. Mice were infected with actively induced encephalomyelitis, which causes inflammation and damage to various parts of the CNS and myelin sheath components of nerve fibers. Results of the study found a clear evidence showing that via the administration of high levels of CBD, the MRI results showed a clear decrease in the effects of encephalomyelitis as well as a reduction in the damage to myelin sheath and its subsequent nerve axons. CBD demonstrated that it had the ability to reduce overall viability of encephalogenic cells as well as cause early apoptosis of such cells, contributing to the overall therapeutic effect generated by the administration of CBD. Similar to the previous two studies, another study also looked at the role of cannabinoid receptors and their influence on various components of health by also administering high levels of cannabinoids upon mice as test subjects. The body has two main cannabinoid receptors, CB1 and CB2, as discussed before, which are the corresponding receptors within many mammals which accept the cannabinoids contained in cannabis. CB1 was found to correlate with mainly the psychoactive effects of cannabis use, which many users associate with the high from smoking. CB2 receptors, however, were found to play an important role in the immune system of the body, affecting the mechanisms which are responsible for activating inflammation as well as various other cell-to-cell communications and protein activations. CB2 is the key receptors which is mainly responsible for medical marijuana's therapeutic properties pertaining to neuroinflammation as well as positive immunomodulation. The study found that CBD cannabinoids are found in high concentrations in the lymphatic system as well as evident interactions with immune system cells, promoting homeostasis within the body via the activation of CB2 receptors. These three studies have comparable outcomes, and the end results in terms of identifying immune system changes due to the administration of cannabis upon the subjects. The shortfall of these studies, however, is that although the subjects were mammals, none of them were humans. This was done to maximize the results from the cannabis dosages in order to easily observe the consequential effects on the smaller bodies of rodents. In addition, because rodents also have much of the same cannabinoid receptors as humans, the results of these studies can be used to get a better understanding of the immunomodulation pertaining to cannabis administration on humans. These studies, however, can only speculate the exact effects of cannabis administration to humans, although it does give researchers grounds to conduct further investigative studies to check the viability of cannabis for treating MS, which is an autoimmune disease within itself. Immunomodulation from cannabis may have benefits for MS as it may help achieve better immune system function as well as act as a protective cover for the myelin sheath covering the nerves of the body. Through this information, it is a viable hypothesis that the administration of cannabis may help prevent further damage from the MS as well as mitigate its existing symptoms. To check if similar results of the effects of cannabis can be seen in the human subjects, it is necessary to run a study using randomized control trials to test the viability of cannabis extracts as a treatment option for patients suffering from MS and its various secondary conditions. The results of the study will determine the efficacy of high CBD RSO oil on various aspects of an MS patient's health. The design of the study itself will be similar to the randomized control trials performed in the studies discussed earlier. However, instead of rodent subjects, it will use human patients suffering from MS. The study will create two groups from the subjects who would be administered pills 3 times a day for the duration of 2 years. Due to the fact that CBD is non-psychoactive, neither of the groups will be able to identify if they are consuming RSO or a chocolate, effectively blinding the test subjects. To add depth and a greater level of understanding surrounding the effects of RSO oil, the study will use questionnaires regarding physical, mental, and spiritual health, which the subjects will complete at multiple occasions throughout the study. The study will also compare results of the central nervous system MRI images on an annual basis to compare the growth of any MS-related lesions or neural deterioration. This data will be then analyzed to determine the feasibility of RSO in treating MS and its symptoms. Although Canada leads the world in MS rates, it is still somewhat of a rare disease. Therefore, it would be difficult to easily recruit a sizable number of individuals required to perform an effective randomized control trial. To increase subject recruitment numbers, the study will be advertised in various MS clinics throughout Canada. A great example would be the MS clinic located in St. Michael's Hospital in downtown Toronto. This will allow the study to be effectively advertised to the target population only. Selection criteria for the study states that the subjects included in the study will be individuals between the age of 18 to 65 who are suffering from MS that has evolved to the point where these patients are demonstrating clinical signs and symptoms of MS. The subjects must also be free of any non-MS related illnesses which may skew the results of the study. One of the main reasons why seniors or individuals over the age of 65 were not included in the selection criteria is due to the high possibility that multiple diseases or illnesses may be present, giving researchers invaluable data for the study given. Individuals who will be using chocolate pills will be considered the control variable in this case. Individuals who are given RSO capsules will be considered the dependent variable. The questionnaires used to determine the therapeutic effects of RSO which may be having on a patient will be considered categorical variables for the purposes of the study. The intervention group will receive high CBD RSO oil capsules loaded with 0.25 milliliters of RSO oil. The control group will receive capsules which look identical to the RSO capsules, however they will be using molten chocolate. Due to the effects of CBD seen in mice, it would be feasible that humans would show similar results preventing further damage caused by MS. For the outcome of the study, subjects in the intervention group will benefit from the high levels of CBD present in the RSO oil. As a result, their MS-related symptoms would consequently decrease, having a positive impact on an individual's quality of life. The secondary outcome for this study may be that the mechanism of immunomodulation caused by CB2 receptors activation will encourage the body of the subjects to restore proper function of the immune system, preventing the body's own immune system from degenerating the protective myelin sheath of nerves. The possibility of a statistical issue may exist for this study. One must acknowledge the fact that MS is a very subjective disease, therefore it is impossible to assume everyone on a level playing field, especially with respect to the number of questionnaires test subjects will be asked to complete in the study. Therefore, it is better that the results of the given test subjects are only compared with their previous questionnaires and MRI scans to see if there is a change in the subject's degree of severity pertaining to MS. There have been 0 documented deaths which have been scientifically attributable to the use of marijuana. Therefore, the chance of a lethal outcome occurring for any of the patient's subjects is essentially non-existent. Cannabis and its extracts are generally considered safer for the human body than most prescription and some non-prescription medication, thus eliminating the existence of any sort of ethical issues or concerns with respect to the study. Moreover, the extract being administered in the study is non-psychoactive, so there is no chance that subjects will experience any hallucinations or other commonly associated symptoms related to marijuana usage. In terms of limitations of this research, high CBD RSO will not exactly give the complete picture with regards to the general effects of marijuana on MS. CBD RSO can all be very hard to find, as marijuana strains considered high in CBD and low in THC content are somewhat of a rarity. Strains known as such as the Charlotte's Web, developed specifically for young children, is possibly the only strain known to have high levels of CBD and low levels of THC. This concludes this presentation. Thank you.